Good morning. We're going to get started. My name is Anoja Perera. I'm from Star Wars Institute in Kansas City. I'm here to introduce Dr. Fei Chen, who is a member of the Broad Institute at MIT, and he's also an assistant professor at Harvard at the Department of Stem Cell and Regeneration Biology. His lab focuses on building tools both in genomics and computation to understand tissue organization and how disorganization leads to disease. He has won many awards during his career, and he has developed really amazing tools for our community. He co-invented expansion microscopy during his doctoral research. In 2019, he published Slide-seq, which really changed how we do spatial genomics. This is when I actually got to know him. Fei was a wonderful collaborator to me. His lab and him had us come to Boston. He showed us how to do Slide-seq and gave us an early head start to get into this field. He cannot be here in person because of a family emergency, but it is my absolute pleasure to introduce Dr. Fei Chen to give this presentation today. Thank you.
Thank you for that great introduction. Can you guys all see and hear me? Can you guys hear me and see my slides?
Fei.
Hello? Can you hear me and see my slides?
Yes, everything's great.
Okay, great. Okay, thank you again for that introduction. And I apologize again for not being able to be there in person with you all. But I'm excited to share our work with you on how we're studying cellular communities and tissues with genomics. And I'll just start out with some disclosure statements. I'm a co-founder of Curio Biosciences. It's a company that's was formed to help disseminate some of the reagents for Slide-seq and slide tags that I'll describe today. I actually no longer have any financial conflicts of interest with Curio Biosciences, as they've been acquired by Takara, and I believe they're at the conference with you today if you have any questions for them. Okay, with that out of the way, let me just preface this talk by saying I think we're kind of in a place of revolution for genomics and I think for pathology and looking at tissues in general. But if we take a step back and think, this is an article that was published by Eric Lander as a perspective in science right before the human genome was sequenced about what genomics kind of meant. And he outlined a couple of different ideas for what we could do with genomics. This is almost exactly 30 years ago, 29, I guess. And they've only sequenced 1% of the genome. And they go through like looking at DNA and variants and stuff. And they have one little section on RNA where there's some predictions that are made, where maybe simultaneous monitoring of the expression of all genes, this is what it says, the mRNA sensitively reflects the state of the cell. So maybe we could imagine monitoring RNA. And then there's one part in that about spatial localization of gene expression. This is about development, but in general might hold critical clues about function. And it also says DNA arrays would not be suitable for such investigations, but maybe in situ hybridizations can. And so, I think in many ways this article was quite prescient, but it also reflects maybe the evolution in genomics and revolution in genomics, because now today we're kind of in a magical time where we can go and look at actually with DNA arrays, all the transcription of every gene in the genome with single cell or very high resolution. And this also reminded me of a little bit of, I'm a big science fiction fan, and maybe some of you guys know this, but there's these famous 3 laws that Arthur C. Clarke put into one of his books. And I think many know number 3, any sufficiently advanced technology is indistinguishable from magic. But I think these first 2 might really apply here in this context. All right, so enough of this digression. But as many of you guys know, I think the revolution I'm talking about is how can we start understanding the molecular states of cells directly into tissue cell in the context of tissues. And our view of this has recently been really shaped by this, is that you can think of cells within tissues as having programs that within them that govern their states and their types. And these are concerted expression programs of genes that might define a cell's identity, or it might identify its current function. Maybe a T cell can activate a cytotoxicity program, or it could have a program that's shared between many types of cells, such as like a cell division program, which is reused when cells are undergoing division between different types of cells. And then at the same time, what types of cells and what states they're in is really actually influenced by the context that the cell lives in. And so we think of this as multicellular circuits within tissues that then govern the cell state. And these multicellular circuits actually are what outputs as tissue function and structure. And I guess we've known that for a long time because, through microscopy and pathology, we've studied tissues not in terms of molecularly defined programs, but more in terms of multicellular circuits. Like we can look at the structure and architecture of the tissue and how that is organized and is disorganized in cells and disease. But I think now, today, we might really be able to look with molecular tools to explore how these two things are controlled and fed back into each other. And then I just want to contextualize that maybe with an example in cancer. And, I'm just using cancer here as an example because I'll also talk about it later on in my talk, but I think it also is a great place for applying these spatial genomic tools because cell types and states are very diverse and are very influenced by their environment. And in cancer, we often have some genotype, whether those are driver mutations or other things, genomic alterations, methylation, that cause recurrent evolutionary trajectories of evolution for some intrinsic state of the tumor that actually drives it to become malignant. And that's actually then related to the phenotype of individual tumor cells because each individual tumor cells might have different states that they undergo, whether they're cell cycle states or states that are more like EMT, like a transition to a more mesenchymal-like phenotype for an epithelial tumor, or states that are reflecting stress, hypoxia, or response to the immune system. And that is then, this diversity of states is both driven by the intrinsic genetic states of those cells and then the environment that they live in, including, for example, their interactions with immune cells. And so the big picture question then is like to understand how cells are organized and shaped by their environment. And, in general, we like to think of it in terms of three questions. I think a lot of people are collecting this sort of data. I think it's important to think about what are the underlying questions that you can answer with the data. So I think the three things are, what are the cell's neighborhoods? Where do you live? What are your interactions with nearby cells? And how do you communicate to those nearby cells through signaling? And then there's one other question is then how does that feed back to change a cell state? So given a context, how does that allow you to use different molecular programs? And genomic measurements, such as those measuring transcriptomes and epigenomes, have really given us great handles on cell types and states in single cell. And so we've really been motivated to develop tools to bridge that into the tissue context. And we've been doing that for a number of years now. And in the lab, we've focused on two types of technologies. One is based on spatial capture, where we use barcoded arrays to capture DNA and mRNA molecules. And then basically, when you capture them with these barcoded arrays, you kind of get to know where those molecules came from in space. Excuse me. And the second type of technologies that we've been developing is directly going and using a microscope and carrying out the reactions that you would find in a DNA sequencer on the cell. For example, we developed some approaches to directly sequence the genome. Here you can see like individual rounds of DNA sequencing reactions. This is in a nucleus. Then you can resolve that into the organization of chromosomes. Today, I'll really mainly focus on this idea of using Slide-seq. And I think the main point is that we want to actually capture the sequence information, whether that's the genomic sequence context, the epigenome sequence context, or the transcriptomic sequence context. Because beyond just counting the mRNA molecules, we think that there's quite a lot of context also in the DNA sequence. And so a while back now, we developed this technology called Slide-seq, which was one of the first high-resolution spatial transcriptomics technologies. And so how Slide-seq works is that it uses barcoded beads to form a spatially barcoded array. Basically, you can take these 10-micron polystyrene microbeads and deposit them onto the surface of a piece of cover glass here. This is a 40-millimeter cover glass. And each one of these white circles has about 100,000 of these little beads on them. And they're densely packed. And they're randomly deposited, but each bead has a clonal barcode of A's, C's, G's, and T's. And we use a microscope and sequencing to decode. So this is like one sequencing reaction, and you can see the different colors. They represent different bases, and we go and decode this barcode so that when you actually go and do the experiment, what you're doing is you're taking the tissue and you're capturing it, and the RNA diffuse onto the beads. And when you sequence them, you have the spatial locations of where those RNAs came from. And we published this in 2019, and this was at 10-micron resolution. Subsequently, it was commercialized, and now I think there's quite a number of technologies that allow you to do kind of this array-based spatial capture of mRNA. This is actually one of the first experiments that we ever did with Slide-seq, where here is a mouse hippocampus. The colors represent clusters of cell types as defined by their transcriptome on these beads. And then here's an adjacent serial section showing like the histology and DAPI. And you can see that now we can assign molecular identities to these types of cells. But I told you that there were kind of these different organizing problems of tissue composition, interactions, and differential expression as a function of interaction. So how do we like study that? And so to do that, I think we need to define a cell's neighborhoods, or how are molecularly defined cell types organized inside of a tissue. And then we need to be able to define basically how does gene expression change as a function of that context. And we developed some tools to allow us to do this computationally, called that folks are using. This is on GitHub on a package called SpaceXR. This is an approach that allows you to take single cell references and assign cell types to spatial capture data, and this is an approach that allows you to do spatial differential expression. But I just want to use one of these examples where we develop a spatial DNA capture approach to kind of like illustrate how we were thinking about these questions of how does gene expression change as a function of environment when we first developed these technologies. And so when we first developed Slide-seq, we captured RNA, and we thought that it would maybe not be too hard to also capture DNA. And there we used the same arrays, and instead of capturing RNA using poly A, what we did was we took tissue and we tagmented it with TN5 transposase, allowing it random DNA in the genome to be randomly cut and ligated to adapter sequences. And these adapter sequences could actually then be captured by the beads. So now instead of capturing mRNA, you're capturing DNA. And in a parallel, it shares the same bead indexing, so you can actually capture the RNA in just an adjacent section. And so this is, we developed this to get a sense of like how does the cell intrinsic state influenced by its environment if the cell intrinsic state is something defined by the genome of a cell in cancer. And just to validate the technology, we first kind of ran it on some metastatic clones that were injected into mice. They were cell lines that you could inject, and they would form liver metastases. And so here you can see a piece of liver, and here's an H&E of these two tumors, and they're actually seeded from individual cells. So they have different clonal origins. When you run this sort of DNA measurement, you get each bead captures the genome unbiasedly. And here what we did was we ran principal component analysis on just like the fragments captured per bead across genomic bins. So the features were just like every piece of the genome segmented into 1 megabase bins. And you can see that PC1 captures one clone, and then PC2 captures this other clone, separating them from normal. But what does that actually mean? It turns out that basically what these PCs pick up on are different. Like if you look at the loadings, they're basically the chromosome alterations that are present in these clones. So for example, PC2 is actually picking up strongly on this chromosome 6 amplification in this Q arm, and then chromosome 15 amplification is strongly picked up by PC1. These actually have a shared chromosome 19 alteration across these two clones. And so simple principal component analysis on the capture across the genome can separate clonal variation, at least in terms of chromosomal number alterations. And so what we can then do is we ran this on a human adenocarcinoma, colorectal cancer. And so here you can see a high resolution H&E separate. And here is this Slide-seq data where now PC1 is actually separating normal cells, stromal cells from tumor cells, and PC2 is separating out a distinct subclone from the parental clone. And this distinct subclone is actually really highlighted by a chromosome 20 gain. And they all have strong chromosome 8q gains because this is a MYC-driven tumor. And you can see that this actually matches up in the histology where the MKI67 corresponds to the regions of high tumor cell density. Okay, so now going back to this idea of like how does a cell's extrinsic environment affect its intrinsic state usage, we performed slide RNA-seq, basically just like normal RNA capture on an adjacent section here. Here it's colored by the cellular cell type identities, whether they're basically like tumor cells, immune cells, or stromal cells of non-immune nature. And here are these clone labels for regions where there are high tumor cell density. And as I said, there were these two genetic clones, subclones separated out, even in this 3 millimeter region of the tumor. And we actually sought to explain the extrinsic factor. There are many ways to explain the extrinsic factor environment of a tissue. We did something very simple in this analysis, but later on I'll tell you about how we're thinking about more complex ways of defining the extrinsic environment. But here we just looked at a simple factor like local tumor cell density, and we tried to explain the gene expression within tumor cells, one as an intrinsic clonal assignment, what clone did you come from, two as a neighborhood expression versus of how many like locally what the local tumor cell density is. And we did an analysis that sounds complicated, but it's actually quite simple. It's something called variance decomposition. And basically what it is that for each gene in the genome, you can ask like how much its gene expression variance is explained by this variable of local tumor cell density, which is like a cell extrinsic factor, or this variable of whether it's in clone 1 or clone 2, which is a cell intrinsic factor. And so that's percent variance explained by clone and or density per gene. So then you can see that for some genes like PLAG1 or FAM3D, it's just quite highly explained by what clone you are. And you can see that here, PLAG1, most of its variance is explained by it being clone 2, and then the opposite for PROM1. And but for other genes, they don't seem like it's separating along the clonal boundaries, but instead it seems to be separating along what the local density of tumor cells is. And so if you look at then what the gene sets associated with these are, the subclone associated genes tend to be MYC targets, and we think that's because there's this differential amplification of MYC between these two subclones. But more interestingly, the density associated genes, basically this extrinsic environmental explanation, seems to be explained by cell adhesion molecule and cadherin molecule binding. And so maybe there's this external program in the cell and nearby cells that cause the local cancer cells to be more dense. And this is not at all related to, this program is not actually intrinsic to the clonal identity of the tumor cell. And so this is just one example of how we're trying to just decompose transcriptional programs into like clonal and environmental compositions. And as I said, like, here we're trying to decompose tissue organization. We map the cell types and states, and then we try to explain differential expression as a function of tissue context. But this is actually quite challenging to do in these capture-based methods. And actually that's because these capture-based methods, including Slide-seq, suffer from cell mixtures where, if you imagine these gray circles, black circles are the beads or pixels on your array. You can't exactly control, even if your pixels are smaller than the cell, you can't exactly control what pixels your cells land on, and thus, individual pixels can be mixtures of transcriptomes from individual cells. And basically what we developed are many tools like CSIDE and RCTE that allow you to overcome this mixture problem. And it turns out that the mixture problem isn't significantly better when you use smaller pixels because, at some point you have to aggregate pixels, and you still have to then think about how to aggregate them in the presence of mixtures. So a couple years ago, we thought to maybe try to solve this problem in a different way. And the idea is that, in this first set of experiments, how I describe these data being collected is we would collect single-cell sequencing to get some ground truth, to get some ground truth cell types and states, and then we would collect spatial transcriptomics data where we would then leverage the single-cell data to actually deconvolve the mixtures. And we thought that maybe we should unify these two worlds. And there's a lot of power in doing that because in single cell, we have really high quality single cell measurements. And these tools have become quite mature and they're getting quite cheaper. And there's an extensive toolbox for a lot of other modalities, including DNA, attack, methylation. But just unfortunately, there's no spatial context. And spatial transcriptomics profiling, has spatial context, but doesn't get to take advantage of many of the existing tools that single cell has already made. So what if we could get, have our cake and eat it too, whereby spatially barcoding the cells, we can recover the spatial information and at the same time get all of these benefits of single cell. And so we developed this approach called slide tags, which really is just running it's kind of like running Slide-seq backwards, but it allows you to get single cell data and know where the cells came from. And so how it works is, again, you take the same barcoded arrays, and now you put the tissue on. And if you remember, each bead has a unique barcode that you know the XY location of. And now instead of capturing the mRNA onto the tissue, onto the beads, we photocleave these beads and allow these barcodes to diffuse into the tissue. And these barcodes then associate with the nuclei of the cells, actually in like a non-specific but quite robust manner, probably due to the charge of the histones. Then we isolate these nuclei, and then you can go and profile these nuclei on established microfluidic or other single-cell platforms. A lot of the work that I'll be showing you was done on 10x. And so what does the data look like? This is our our actual first experiment, one of our first experiments with slide tags. And again, we did it on the mouse hippocampus because there's a lot of both cell type diversity as well as spatial architecture. And so here you can see a UMAP representation of all the different cell types in the hippocampus. You can see quite finely grained cell type coverage and diversity. But now if you look at a given cell, you can see that this green cell might have many, many spatial barcodes that it captured. So now this is like which bead barcodes did it find? And you can see that it captured many barcodes at x equals, let's say 1,000 and y equals 1,500. And here's actually like a distributional plot. And you can see very easily that you can fit the peaks of these distributions to localize the position of the cells with quite high accuracy. Actually, this is like a form of error correction where the distribution allows you to be robust to any given spatial barcode. And so when you do that, you can assign spatial placements to the majority of these cells. Here's just a cool animation showing that you can do that. And underlying each one of these cells is actually the gene expression profile of a true single cell data. And so you can then plot individual genes back into space. And I think the key thing to take away here is that this spatial barcoding is an upfront step that takes like 10 minutes before you do your experiment. And then you do your single cell or single nucleus experiment the same way, but it doesn't affect your single nucleus RNA-seq quality. So here you can see on basically serial experiments where we ran normal single nucleus RNA-seq or slide tags, you get the same cell type proportions, the same number of mean molecules per cell, thousands of molecules per cell, same normalized average counts for a given cell type. And that's just because it's the same data. And then, I think the only thing to say is that, you don't get every single cell that you sequence from the tissue, right? You lose some cells in the drop-up barcoding, you lose some cells in the nuclei extraction. In the first version of the protocol, we estimated we're getting about 10% total recovery from the tissue. And we've made some improvements to the protocol where we are actually getting probably 25 to 30% of the nuclei from any given tissue spatially mapped into space. And so this is, we think, given the quality of the single cell data, we think this is great. And we've done some downsampling analysis on both receptor ligand interactions or niches to show that these things are robust at these levels of recovery. Okay, and so one of the most exciting aspects of slide tags then is its compatibility with existing nuclei sequencing as well as analysis tools. And so that includes maybe like RNA expression, which is the workhorse of most single cell technologies. But if you think about it, there's ready ways to get TCR sequences, copy number inference, chromatin accessibility. And so in our paper, we highlighted a number of different examples where we did this with different tissues and different analyses. But I'll just highlight one example where we applied it to a sample of metastatic melanoma. And I want to just highlight this example because it shows kind of the versatility to do a lot of these other different single cell approaches. So we applied slide tags to a human lymph node metastasis. This is after checkpoint blockade. And I'll just walk you one sample where we actually see recurrent conclusions from many samples. So in this sample, we see that the H&E revealed two distinct histological regions. It's actually separated here by this dotted green line. There's this fibrous stroma that's separating two tumor regions. And then when we apply slide tags to it, you can see that we recover the majority, quite diverse cell types and states, including many diverse stromal cells and immune cell compartments, as well as some diversity in the tumor cell compartment. These three distinct clusters, we can map these back into space, into the spatial organization of these cells directly. And then if you look at these tumor cell compartments, they're each defined by unique transcriptomic states. When mapped spatially, these actually segregated into spatial regions. They actually align quite closely with these two regions that we identified, with tumor state 2 being in one region and then one A and B being on another region. Next, we wanted to understand if these transcriptional states actually are driven by genetic differences. So then we used the genome-wide data and we used copy number variation across single cells. This is shown on the rows. Each row is a single cell. And with red actually showing the gains. So columns represent positions along the genome, numbers representing chromosomes. So you can see that there's some shared gain of chromosome 7 and loss of 10. And then suggest this is just a common ancestral clone, but then there's some unique changes that reveal these two genetic subclones. This tumor 1 showing additional transcriptional heterogeneity, it has this loss in chromosome 6. Probably, it looks like from the data, this is like a subsequent clonal branch from state 2. So we showed how these tumor cell states are spatially segregated. So then we want to understand if they have like different neighborhoods. Do they have different cellular environments? And so we did a simple analysis of basically co-localization or enrichment of immune and stromal cell compartments. And you do indeed see that basically clone 2 and clone 1 have different states, actually even 1A and 1B. And in particular, red shows the enrichment and blue shows the depletion. You see that this clone 2 has a strong enrichment of CD8 T cells, and clone 1, at least 1B, seems to have a strong depletion. And so CD8 T cells in particular were enriched near one tumor clone, which actually piqued our interest. And so we then performed TCR sequencing using, here we actually used 3' 10x, and we enriched out the TCRs and did long-read sequencing. But these days, more and more, we actually just use 5' profiling that directly allows us to access the CDRs. And so then mapping the CDR clonality, we found a very interesting thing, which is that it revealed this one subclone here. That's this one subclone shown in purple, is highly expanded and is interacting with this tumor 2 subclone. That's actually driving the majority of this enrichment. The gray dots represent all CD8 T cells, and the purple dots, all other CD8 T cells, actually, I would say. And the purple dots represent this particular clone. And so you can see, even without any statistics, that it's strongly enriched in this one, with this one tumor clone. And so, so this is like, for the first time, we were actually able to observe CD8 T cell clonality linked to specific genetic tumor subclones. And, this shows that maybe the tumor cell lineage itself has actually played a major role in shaping these interactions. And so how is it doing that? Well, there's probably 2 ways that it can do that. Well, one is that it could, this tumor 1 state could be losing its melanocytic identity and associated with endogenous antigens. And so melanoma has this dedifferentiation phenotype where it can become more neurocrest-like or mesenchymal-like through this dedifferentiation trajectory. And when we performed, we actually went and performed joint attack and RNA sequencing on this sample. And you can see TNC, which is a previously identified marker of this state transition, has broad chromatin accessibility as well as broad expression in the tumor 1 subclone. And so, you can actually test this hypothesis a little bit more by, we score tumor cells on the expression of mesenchymal-like state genes. And you can see here that tumor 2 has a very low expression of these genes, and tumor 1 has a very high expression of the score. And basically what's happening is in tumor 2, in tumor 1, you're downregulating PML and MLNA, which is this melanocytic-like state, the epithelial state, and then upregulating these TNC and AXL expression, which is associated with this mesenchymal-like transition. And so, so we actually looked back to our DAC data in the same nuclei to see if we could identify transcription factor motifs associated with this mesenchymal-like state. And so here in this plot, on the x-axis is actually correlation with this state for different transcription factor motifs. And you can see that FOS, June, and IFR9 are positively correlated with mesenchymal-like scores. This kind of makes sense. It's previously been shown that these immediate early genes or stress response genes are correlated with this mesenchymal-like transition. On the other side, MITF, this is the master regulator of the mesenchymal-like state, is downregulated in the Steve differentiated state. More interesting, on the y-axis is like we're mapping these TF motif scores in space, and you can see that on the y-axis is basically how spatially correlated these different motifs are. And you can see that basically some of these TF motifs are correlated spatially in this mesenchymal-like state, or clustering spatially together. And this is maybe suggestive of tumor microenvironmental pressures or maybe epigenetic inheritance through cell division. Okay, so just to summarize this first part, we discovered significant transcriptional and clonal heterogeneity in tumor cell populations in melanoma, actually corresponding to distinct spatial communities. And then we found that specific TCR clones are actually associated with specific tumor subclones. And one thing we're trying to understand is, like, how does this intrinsic tumor properties actually influence immune interactions? And I think this opens up this question that a lot of us are quite interested in, is like, are these interactions primarily driven by intrinsic cell states of the tumor or cell intrinsic environments? And in this case, it looked like that tumor cell clonality played like a dominant role in modulating its local environment. And so we're starting to look at that more systematically. And so that's this coming back to this beginning of this talk, which is like, how do we relate the programs inside of a cell to the environment outside of a cell? And so we've been, because of these interesting observations in melanoma, we set out to collect a much larger data set of slide tags in melanoma. We actually collected across 300K nuclei across 27 metastatic pulp biopsies. This is a collaboration with Nir Hacohen. And here, in this data set, this is all pre-treatment, although we do have response status for these patients. But we wanted to actually then use this data to understand if there were recurrent neighborhoods in these samples, and then also how do these recurrent neighborhoods affect gene expression. And so first we asked, like, can we identify basically niches which associate with tumor cells? And so here we took all of the non-tumor cells and we clustered them by basically their spatial interaction with each other. And we're calling these niches because they're basically the neighborhoods of cells which co-associate with each other. And if you look at this, this first niche is something like we're calling an immune effector hub. This is kind of a recent observation by many, including Nir and others, of populations of macrophages, DCs, and kind of tumor reactive T cells, CD8 T cells, that form local structures within tumors. There's this interesting myeloid/stromal niche, which I'll talk about in a bit. There's something that looks like TLS, like a tertiary lymphoid structure, with CD4 T cells, dendritic cells, and B cells in there. So some unknown, actually, niches of an immune cell population, some vaster niches. Basically, these are kind of, across all of these samples, constantly reoccurring structures. And you can kind of visualize them. So here in this sample, this is the immune effector hub kind of infiltrates into the tumor, while at the same time, there are some tertiary lymphoid-like structures at the boundaries, and also over here. And so then we want to ask, we set out to ask this question, so now we can define cellular neighborhoods. Then how does this change gene expression within cells? And there's many ways that you can do this. One way is you can perform simple differential expression. For example, you could ask, here in malignant cells, as a function of in proximity to these cytotoxic T cell neighborhoods or far away. So here is like nearby. These are cancer cells near the cytotoxic T cell neighborhoods, cancer cells away. You can see near the cytotoxic T cell neighborhoods, you have basically interferon responses, as well as TAP. That's like an interesting presentation. And it's, so this is one way you can just do differential expression. We thought maybe there would be more principled ways of doing it by first identifying the gene expression programs within the malignant cells. So, within the tumor cells, we might be able to identify recurring sets of genes that they use across all the samples that define their states. And so we did that. So this is a heat map, but basically this is a heat map of like the correlation between genes, gene usage across different tumor cells. And these represent then the programs that these tumor cells use. And then you can identify what these programs, basically we can go and manually annotate them. Some of them represent like proliferation. Some of them might represent like this EMT neurocrest, which is like this dedifferentiation trajectory. Some represent stress apoptosis, interferon response to an immune system, the immune interferons. And so then we can ask this question, like, we define all these cell programs within the tumor cells. How does what neighborhood you're in affect what programs you use? And we did this analysis that sounds hard, but it's actually quite straightforward, which is a linear regression, where for each malignant program, we asked like which cell types in the stroma are most predictive of that malignant program's usage. And so there's a lot of entries in this matrix, but basically I just want you to pay attention to these two, there's basically 4 quadrants. There is this quadrant up here, and this is like many cells that are upregulated in their interferon response. This is M17, and they're very just like what's predicting that is their interactions with many of these basically tumor responsive cytotoxic T cell neighborhoods. And as well as this macrophage cyclic 10 population. So CD8 cycling, tumor responsive CD8s, and this macrophage population. So you can actually see here that this cyclic 10 macrophage population is highly correlated with this interferon response program. This is in tumor cells, and this is the macrophage spatial density. There's this other SPP1 macrophage, which I'll get to in a second, which is anti-correlated with this interferon response signaling program. On the bottom here, this other block is anti-correlated with this interferon response, and these are all these neurocrest dedifferentiation programs. And they basically deplete these reactive T cells, and they're highly enriched in a lot of this MAC SPP1, the suppressive macrophage population, as well as this fibroblast post-N population. It's an interesting fibroblast population observed in other tumor types that maybe also be immunosuppressive. And so you can see that here. So there's this, if you just look at basically this is like the scores that explain this program usage, and MAC SPP1 and fibroblast post-1N are highly correlated with this program usage. You can see that usage of this program here, and then the MAC SPP1 populations here. Okay, and so actually, so what's happening is that there are these two distinct macrophage populations, and it seems like there's this suppressive macrophage population that's occupying this myeloid/stromal niche with these post-N fibroblasts, and it's also predictive of this neurocrest signaling program in malignant cells, which we know is this dedifferentiated state. And then there's this MAG cyclic 10 plus population, which is occupying these effector immune cell niches. And they're co-localized with basically the interferon stimulated and dying cancer cells. And so this is like what you want to have. But you notice it's not across tumor, we see this across many, many samples, but it's not that you see the same populations actually in the same sample, even in small space. This is only like 6 millimeters across. So across different regions of this tumor, there are different tumor cell states, and they're actually have different environments. And we also find that these populations are actually highly predictive of immunotherapy response. So it's very possible that upon immunotherapy, you actually get some selection of these different environmental populations. So not only are these macrophage populations separating out these two niches, we also seem to see that they're associated with many soluble ligands, including like different interleukins and also chemokines. And so we wanted to see if these soluble ligands are actually basically the molecules that are mediating the communication between cell neighborhoods and cell intrinsic gene expression states. And so we've been thinking about a way to define cell extrinsic signaling environments in the same way that, we've been defining cell intrinsic gene expression states and cell neighborhoods. And this is actually, what we're doing is that we can take, for any given cell, we're just looking at all of the basically soluble signaling ligands in proximity to it. We build a matrix of, all of the soluble signaling ligands that cell has observed. So a vector, right? And across all the cells, that's a matrix. And that's cool because it's almost like a gene expression matrix, but it's like the gene expression matrix of the external signaling ligands. And then you can decompose that matrix very, very easily using things like NMF or other factor decomposition programs across all of your samples. And that gives you these recurrent covariant signaling programs. And so when we do that, we can start to finding some of these signaling programs. So here are 3 signaling programs. It turns out that basically these 2 signaling programs are associated with these 2 tumor populations, what we called this neurocrest-like program. This dedifferentiated state actually has like a lot of neurosignaling ligands as well as like Wnt and PDGFA. There is this more like immune and also like signaling program that's associated with the MITF melanocytic state, probably related to the fact that that's really quite enriched in the immune effector hubs. And then this thing, CXL9, 10, 11, actually defines a signaling program that's responsible for the creation of tertiary lymphoid structures. Or maybe not the creation, but the steady state observation of tertiary lymphoid structures. And so this is how we're thinking about like spatial data actually in general. So like we think that there's basically recurrent spatial neighborhoods that define stromal and immune cell circuits, especially in cancer. And this is if we can observe these recurrent neighborhoods, then we can go and use that to explain context-specific gene expression changes within cells. And then what's mediating this communication is like extracellular signaling programs. And maybe we can start, instead of defining single genes, receptor ligand interactions, let's look at like the recurrent set of programs that reoccur across many tissues. And so that might be more informative, just like how that's been more informative for us for gene expression. And looking at these things, they're going to be important for like learning the rules of tissue organization, basically like how our tissues developmentally patterned or maybe also pathologically patterned, as well as for therapy and engineering of tissues, because we like to then be able to manipulate these neighborhoods. Okay, so I just want to summarize the Slide-seq technology, and then I'll have 2 quick future-looking vignettes about the technology. So Slide-seq allows you to do single cell with high spatial resolution, but the spatial resolution is the resolution that you're placing the single cells, like not the resolution that you're capturing the mRNA. So like the data is single cell resolution, it's just you're replacing the cells with about 10 micron resolution, which is about the size of a cell anyways. It's compatible with existing single cell workflows and analysis. And then the answer is like, I think we should be able to do it then for every type of single cell experiment and assay. And one assay that we've been quite excited about doing it for is DNA, because we care quite a lot about, like, as you saw in the previous talks, like the intrinsic clonal evolution of cells. And so we've been, I talked to you about Slide-seq. I think it's pretty good for CNV calling, but it has pretty low spatial resolution. So single cell requires deconvolution, and the per cell coverage is actually not that high. And so then you can't look at single nucleotide variants. And so we've been modifying a couple of different technologies for slide tags, single cell technology for slide tags to capture DNA. The first is a droplet-based technology where basically, this is off the 10x multi-omic kit where we do the slide tags protocol as normal, and then we get the nuclei. But before we tagment them, we treat the histones with a slight denaturant, and this allows the chromatin accessibility to be lost, so then you get unbiased coverage across the genome. Here we did this on like a human colon specimen, normal human colon. You can see basically placements of individual cells, as well as for each cell, we're getting about 1 million fragments or 600,000 fragments per unique fragments per cell. If you do the back of the envelope, that's something like 2 to 3% coverage for every given cell. That's perfect, actually quite overkill for CNVs. I think for SNVs, from individual cells, it requires a lot more computation and aggregation over nearby cells. And you can see that, obviously, it's also good for mutational signatures. Here you can see the resolution of mutational signatures, including SBS5, which is the primary aging signature for this tissue. And here I'm showing you one single nucleotide variant. There's like 10,000 single nucleotide variants in this sample with variant allele frequency less than germline. So this is just one of them. But I'm showing you this one because here's the colon, and you can see the individual crypts. You can see the layers of different cells, and the crypts are oriented this way. Here, this variant is actually spatially clustered, where the variant allele is like all clustered in one region. And we observe many variants that are like that, actually. And actually, these variants probably correspond to these epithelial crypts as we'd expect. And with the right computation, I think we have the hope of actually re-aggregating across types of cells and spatially localized cells to actually get local lineage information, which is pretty cool. The other thing that we've been doing actually is using it with Bioscribe. Has this single cell genome and transcriptome protocol that's in plates. So it's obviously a lot lower throughput than droplet-based approaches, but on each individual cell, you can get like 80% coverage of the genome. And so this is one of our first experiments where we just sequence like 8 cells, actually, or I don't forget how many, but maybe a little bit more than 8, but on individual plates. Each one of these represents the number of spatial barcode UMIs on the array. And you can see like each individual cell has like very distinct localization. And then the idea is now we can actually start sequencing the whole genome, and we can use that to spatially like assign clonal fields in situ when we reconstruct phylogenetic trees using single nucleotide variants. Okay, so that's DNA. I also want to say that I just think that we should be doing this sort of barcoding in front of every single cell experiment, because it doesn't change the data. And so if you're already running single nucleus RNA-seq on tissues, you might as well get the spatial context for free. And I just, single cells has seen this immense scaling in cost and increases in scalability, probably something that you guys are seeing at your conference. This is even a pretty old plot. And so right now, like, as single cell costs come down, then what's actually the bottleneck to doing this sort of thing on every single cell experiment? Well, one thing is it's been really bottlenecked by experimental throughput. This is a cool Stereoseq data set that was previously generated where they looked at a whole embryo. Here, like in humans, ideally we'd like to be able to do whole embryos over time. Human cell atlases, the human brain is about 9 centimeters in size. This is like a cortical section, or many case control specimens in human tissue, which are much bigger than 3 or 6 millimeters. And so both the scale and the size of arrays and the number of arrays has actually been kind of a limiting step. And that's because we, in our group and also in these commercial technologies, like you have to go and produce these arrays. And that on the microscope, that is actually takes time and is challenging. And it requires some specialized equipment. And we actually open sourced. When we first published Slide-seq, we open sourced the protocol to reproduce it. And some groups actually reached out to us to try to open source it, to reproduce it. And basically, like, the effort was very high in that only one group actually ended up doing it. And that's, a big motivation for us setting up, helping, share the technology with Curio. And they've done an amazing job in disseminating the technology because they've, they're great, automated the manufacturing of these arrays. But if we want to go and make really large scale arrays for thousands of experiments, we thought that maybe there still needs to be ways where we can really scale that. And so we had this idea that, it was a little bit inspired by slide tags, that, if you look at each one of those individual nuclei, they captured something about the local neighborhood of spatial barcodes. And there's been some thought in the field that you can reconstruct the spatial information without doing any imaging. The most obvious thing is in high C or proximity interactions, right? Or CLIP, you can directly know what molecules are next to each other based on the junctions or sequenced reads. And if you get enough of these observations, you can try to actually reconstruct the architecture of the genome, at least ensemble, through the interactions. We know actually also that if you have a map of the pairwise distances between all of the US cities, if you have a list of the pairwise distance between all US cities, you can easily generate a map of the United States, actually. And so we thought that maybe because we were already observing that each individual nuclei was picking up kind of the diffusion of all the beads underneath it, this diffusion is a little bit equivalent to a pairwise distance measurement, basically how far away this bead is from the nuclei. And so can we then basically from our sequencing data reconstruct the spatial data? And so that's the inspiration from slide tags. How we implemented it was a little bit different because we wanted this sort of diffusion to be more controlled and uniform across the array. And so we did a simple experiment where we took these beads and we mixed them 1-to-1 with 2 different types of beads. One type of bead was a capture bead that has oligos that have poly T on them, and they're not photocleavable. And another type of bead is a fiducial bead, which are photocleavable and has poly A on them. If you note that these beads that are photocleavable can be readily used for slide tags because they would diffuse into the nuclei, and then they could be used as, with poly A priming. When you do this reaction, and basically what happens is these guys go over and get captured locally by the nearby beads, when you sequence it, you end up with this matrix of barcodes in the capture beads by how many barcodes it saw in the diffusion beads. It's like basically almost like a single cell matrix, right? And the idea then is that given this matrix, you want to know for all of the barcodes, what was the XY location? And there's this very interesting thing where if you think about it, this matrix is a high dimensional piece of data where the number of dimensions is the number of beads. And like this is a number, you can imagine if this is a cells and this is the number of beads, this is the number of beads, is the number of dimensions. It's actually very similar to a single cell experiment where each cell is sampling genes in 20,000 dimensional space, right? And so in cell by in genes. And often we use dimensionality reduction to visualize this high dimensional data in 2D because we believe that, cells that are similar in high dimensional space should have similar gene expression, should be clustered together in the 2D embedding. Here, we have a real 2D surface that has generated the data from a physical process. And thus, there's a low dimensional representation should be able to capture the actual physical properties of the data. And so, we just applied standard dimensionality reduction on this matrix to 2 dimensions, actually using UMAP. And so then you can then use these arrays for both. And so we ran this on Slide-seq. And so here is just, we basically explored many UMAP parameters. And we colored this array, this rainbow, based on the locations of the beads. It should reconstruct to a rainbow if it reconstructed properly. But we actually also have the ground truth locations. And so here we performed, this is Slide-seq data performed on this reconstructed array on mouse hippocampus. By eye, it looks, pretty reasonable. But, how do we actually characterize the error? Well, we actually have the ground truth data, and we can measure for each bead where it was in the reconstruction versus the ground truth. But the measurement that we actually should care about is, here's the ground truth and here's the reconstruction aligned together. And you can see that there's some deformation that's nonlinear, but local. And so what you actually care about is the error in the length measurements between any pair of points. If you were to measure the length of the hippocampus dentate gyrus in the reconstructed array versus the ground truth array, what's the difference in the error? And so that's what this plot is showing. As a function of measurement length, the error in the reconstruction starts out at about 10 microns. You can't really do better than 10 microns because the size of the bead is 10 microns. And, is maximum at 20 to 30 microns. It's about 2 to 3% of the measurement length, actually. And what this actually means is that the data is virtually indistinguishable in this reconstruction approach. And you can just kind of see that because if you just look at the ground truth versus the reconstruction, oftentimes you cannot see the difference in the data. And if you make measurements, for example, the width of CA1 between these 2 data sets, there's really no difference. And so what does this local deformation actually represent? It's represents a little bit of stretching, or basically it's a little bit of stretching of the tissue or shrinking of the tissue. And the order that this happens is about 2 to 3%. And it's actually quite similar to the level of histological stretching or shrinking that you would get from serial sections. This is actually 2 serial sections we took on a cryosection of frozen tissue. And that kind of makes sense because it's about 1 to 2% of the tissue. And so basically, we think that the error is basically like, you know, it's basically the data is basically equivalent in the reconstructed form and the ground truth form, and you can see that here too. And basically, this is like measurements of pairwise neighborhoods in these two data sets, and again, they're virtually identical. And so then we performed this on. I see. I should be wrapping up now. So we performed this on a P1 mouse brain section. And so this is actually showing the UMAP epochs. And you can see how the algorithm actually then unfolds and reconstructs this piece of data. And this is on a 1.2 centimeter array. And we can reconstruct the majority of cell types and their marker genes, very high resolution. Even these thin layers of cell types are represented by their marker genes, including these actually very fine layers in the olfactory epithelium that have been previously characterized to represent cell type. These are like different cell type boundaries in the epithelium. And I'll just wrap up by saying that we can scale this up by to arbitrary sizes of arrays. And so we did 6 millimeters, 1.2, and now we've actually done some 7 centimeter arrays. This is really led by Evan McCosco, actually, for the BICAN. And this is about the size of a human brain. And you can actually see here is a human brain slab that was here. This is actually a human brain. And this is the basal nucleus and 7 centimeter array. And here's the reconstruction. Here's actually Allen histology. But now we can resolve many different, spatial resolved and substructured cell types on the 7 centimeter data. And so yeah, that's the idea. There's no spatial measurement. You can reconstruct it from sequencing only. And I think this is actually going to be quite powerful because sequencing costs are really quite coming down. And it's going to really enable measurements for every single spatial experiment. And so we're thinking about scaling to whole human organs. We're thinking about spatial and epigenomic genomic and epigenomic measurements. And lastly, something my lab is always quite interested in is how do we infer dynamics information from snapshots, which I think is still hard. Thank you, and I'll take any questions now.
Please come up and use the mic.
Hi, Don Baldwin from Fox Chase Cancer Center. I'm interested in your melanoma cohort. Do you have large-scale characterization, like, for example, TMB status, BRAF mutation driver status? I'm wondering about questions like, are some of the neighborhoods driven by whether they're TMB low or high? Is the entire tumor TMB high, and is it born that way? Does it evolve away from that? Or things like that, where you're trying to integrate normal pathology, molecular pathology, with your neighborhood resolution?
Yeah, that's a really good question. We do have those things. Actually, I think we are trying to look at mutational burden, actually.
Can you call that from the RNA?
You can call it from the RNA, actually. We also have the paired genomes. I mean, not everyone's been sequenced just from a cost perspective. But the cohort is a subset of a pretty large cohort of 200 specimens collected all pre-treatment checkpoint blockade, but wherein we have response status after. And so I think TMB is very interesting. That is something we're quite interested in. I think more interesting is like when we observe different subclones within the same tumor. I mean, they actually have similar levels of tumor mutational burden, but one has just accumulated more CNAs. We often observe that the subclones have very drastically different microenvironments. And and one of those will be immunosuppressive and the other one will not be. And the question, I guess, is that, is that a selection? Is that just like a selection thing, or was it born that way? Or yeah, we're trying to come up with good ways to answer that.
I imagine it's a selection thing. So I'd have two suggestions. One, different microenvironments may participate in releasing circulating biomarkers differently. So it'd be intriguing if you could help us with diagnostic and longitudinal profiling by saying, okay, one microenvironment produces these biomarkers and a different these biomarkers, because a response may be limited to one neighborhood and not the other, and you might be able to see that in the plasma. And then the other suggestion was in your diffusional matrix, you're going after, with poly-T priming, the entire transcriptome, right? What if you used probes that are gene-specific and instead target particular driver mutations or particular biomarkers of a disease? You might reduce the complexity, but still give us pathologically information that we wouldn't require the imaging and be able to do, a higher throughput or a longitudinal kind of thing on serial sections.
Yeah, no, those are both good suggestions, especially especially the first one. We just haven't, we have been looking into some probe-based enrichments on the DNA side, actually, because there the sequencing costs just become quite burdensome. But on the RNA side, we've just been biting the bullet and saying that we can always sequence it to saturation. But I really like this suggestion about the biomarkers. And I also just think the other thing that we're thinking about, we're trying to spend a lot of time doing now these days actually is like, how do we do perturbational experiments to establish the causality? Because you don't really know if it's the selection is going from the tumor cell to the microenvironment or the microenvironment to the tumor cell.
Hey, Dr. Chen, Marcus Casperson out of Mayo Clinic. Just more of a base level question. On that slide where you I believe it was single cell sequencing, where you had about the 10% resolution at the start and you got it up to 25%. Do you believe that you'll increase it to even higher resolutions and that there will be an upper limit for resolutions, or do you think that the sky's the limit in that regard?
Well, I guess you can only ever go up to 100%.
That's fair. Appreciate it.
But I do think there are things to improve. There's some tissue specificness to it. I think we're looking for general protocols that work across many tissues. Probably you can optimize it for any given tissue if you spent the time, but we want to have a protocol that works for many tissues.
Okay, thank you, I appreciate it.
Excellent talk. It's Bonnie here from Yale Center for Genome Analysis. So I have one small technical question. So one of the challenges that we are facing, and when we actually tried to do Trekker, was transportation. So it seems like you have to section the slide, and then you immediately have to process then and there the nuclei, and then then only you can transfer it probably. Yeah. So is there any solution for that? Because the biggest challenge I think for many core facility is that the spatial technology is done in genomics core, while the histology is not integral part of those buildings. So that means you have to transport a little bit. So is there any suggestions for that?
I think so. Well, I don't know. We're testing. So you basically you could freeze the nuclei, or you could fix the nuclei. We're starting to test I guess I think Parse has this, not Parse, Fluent. Fluent has this fixation methanol protocol, I think, for stabilizing the nuclei afterwards that we're starting to test. But I think those are probably the best bet. The other thing that we have tried, although not extensively, is sectioning this thing onto the array and then freezing it at -8, just freezing it then. And then don't do the extraction, don't do the rest of the experiment until later.
Can you actually section and then put some SSC or something and won't allow the slide to dry and transport, something like that? Have you ever actually thought of doing something
I think
like that?
you probably do that, but then you're just worried about like the RNA stability. I don't know how long it'll take you to transfer the thing.
I mean, we are
Yeah.
looking for like 20, 30 minutes, but
Oh
if
yeah,
it is
probably then that's fine. Yeah.
Hi, Elsa Molina from the Salk Institute. So I have a question about the photocleavable spatial barcodes. You showed us on your slide, basically they tag one nucleus per cells, but I was wondering when you have cells with multi-nuclei, how you do?
So they tag like many nuclei. Each photocleavable barcode tags many nuclei, and each nuclei gets many tags so that you can like triangulate effectively like the location of a nucleus. I guess your question might be like, okay, I might have a multi-nucleated cell.
Yes, exactly, yes.
And then I guess you just won't know it's multi-nucleated unless it's like co-localized. I think that's a tricky question. I wonder if you could dissociate it as single cells, or maybe I think there could be other methods too.
Okay, so you haven't tried yet?
I think basically they get treated as separate nuclei, and they would
Okay.
get relocalized back. It's just two nuclei now.
Okay, thank you.
Hi, Fei. It's Johannes from Beth Israel. Great talk. You answered a portion of my question in the end where you showed we can scale arbitrarily large. I think, our next goal is like large archival FFPE tissue that can be, arbitrarily old VDJ on the same slide. And as was asked previously, hotspot mutations by adding specific probes against, something. How close do we think we are? Which ones you think we can push right now, and which ones you think are a bit further away and we should wait for, version 2?
I actually think the DNA is a little bit closer than the RNA for large FFPE. I just think, yeah, on the RNA side, it's just one thing we've been doing is, although it's Evan's really been doing it, is just using flex afterwards, and that seems to work well. So depending on your mileage with flex on those samples, does that make sense?
Yep.
And so yeah, depending on your mileage with flex on those samples, then it is what it is. I think generally it's pretty good.
With nuclei on FFPE, with flex, we get almost perfect results with samples that are 15 years old.
Yeah. So then you can take those really large arrays and run flex on them. I think that would basically get your data. I think the DNA thing is also robust to fixation, although we really haven't tried that much FFPE to know what the fixation artifacts on sequencing, like on mutational calling is. I think that's the main outstanding question.
Thank you. Amazing session.
Thank you very much, Fei. That was fantastic, and that wraps up our session. Thanks a lot.
Thank you.